So in this lecture, we're going to look at smooth muscle. Smooth muscle is found in the walls of most of your hollow organs, except for the heart. Of course, the heart contains cardiac muscle. Example of a hollow organ would be the small intestine here. And so the small intestine would be lined with smooth muscle, which helps push food through the intestine. Reviewing some of our histology, the microscopic structure of smooth muscle. The cells themselves are spindle-shaped, which means they're somewhat elongated, so we do refer to them as fibers, and they're tapered on each end. This shape may also be called fusiform. There's only going to be one nucleus and no striations in this tissue. We're going to have a lack of connective tissue sheaths, as we do in skeletal muscle. We don't have an epimysium or a perimysium around fascicles, but we do have endomysium. So the individual smooth muscle cells will be surrounded by an endomysium. So the hollow organs, like the intestines, urinary bladder, are going to have smooth muscle, even blood vessels, except for the smallest blood vessels. So the smallest blood vessels wouldn't have them. So in these hollow organs, the smooth muscle will be arranged in sheets or layers. One of the layers, the muscle fibers are going to run lengthwise with the organ, and those will be called the longitudinal layer. And then in the other one, the fibers would be arranged in a pattern that goes around the organ, and that would be the circular layer. So when the circular layer contracts, that would cause the lumen of the organ to constrict. Whereas when the longitudinal fibers contract, since the length of the longitudinal fibers run along the length of the organ, like this intestine, when they shorten, they're going to cause the muscle fibers shorten, they're going to cause the small intestine to shorten as well. And so that's true. So in here, the outer layer would be the longitudinal, and if we cross-section through a small intestine like this and looked at the cells in there, because the cells are running lengthwise and we section through them, we would actually be looking at the muscle cells in cross-section in the longitudinal layer. And then since the smooth muscle cells are circular in the circular layer, when we section through there, we would see actually a long section through the muscle fibers. So when these constrict, they basically are going to make the diameter of the lumen, or the hollow within that organ, smaller. And when these two layers contract in an alternating pattern, they create an action that is referred to as peristalsis. So this alternating action of peristalsis can help move items like food, a ball of food, through the tubular organs. So in this case here, you see a bolus of food moving. This could be through small intestine. It could be moving down the esophagus. So when the circular layer contracts and constricts, like at this location, and then when the longitudinal layer contracts, it can shorten that area. And overall, the repeating alternation between shortening and constricting the circular layer would move the bolus of food through the intestine, for example. At the microscopic level, there is no neuromuscular junction like we see in skeletal muscle. Instead, you're going to have the axons, or fibers, of autonomic nerves innervate the smooth muscle. And so these autonomic fibers, the fiber itself, or a neuron, recall that nerves are going to have extensions called axons. The axons, because they're very long and thin, can be referred to as fibers as well. So the axon can be called a fiber. But the fibers in the autonomic nerves are going to have swelled up structures all along there. And you see that here in the image below. As we run down the fiber, you have these swelling areas, and those swelling areas right here are referred to as varicosities. Okay? Those varicosities are going to have the synaptic vesicles that contain neurotransmitters in there. And so instead of calling it a neuromuscular junction, when these varicosities attach to the smooth muscles, they're attached at in areas called diffuse junctions. So instead of neuromuscular junction, it's a diffuse junction, and that would be where your neurotransmitters are released. So we zoom in right here to one of these varicosities, you can see the synaptic vesicles that contain the neurotransmitters. So smooth muscle, though, when we look at it, some of the differences are part of the reason why we don't see striations. There are no sarcomeres. It is the regular arrangement of sarcomeres within skeletal muscle that gives that striated pattern, but we don't have that here. So there are no sarcomeres, and there are no myofibrils. Myofibrils are the structural units of the sarcomeres, are the units that we find within myofibrils. We're also not going to have any T-tubules. Okay? Now, with no myofibrils sarcomeres, you get no striations, but that doesn't mean we don't have myofilaments. So we still do have the myofilaments that are involved in contraction. These are the contractile fibers. They include myosin and actin. So that's the same as in skeletal muscle. It's just that these filaments are not arranged into sarcomeres. So we'll see more of what that looks like here in a bit. Regarding the sarcoplasmic reticulum, the SR, it's going to be less developed than in skeletal muscle. So if skeletal muscle gets its calcium ions from the sarcoplasmic reticulum, and smooth muscle doesn't have as much sarcoplasmic reticulum, then where are we going to get our calcium? Well, most of the calcium used for contraction is going to come from outside the cell, so extracellular origins for that. And so where does the calcium enter? It's going to enter through calcium channels that are located in little pits called calveoli. So if we look here at a picture of or a diagram of a smooth muscle cell, which doesn't have striations in it, you can see the calveoli here labeled. So several of these pits in there, within those pits are going to have the calcium channels. And they get stimulated to open and allow calcium to go in to the cell when the muscle cell is stimulated with action potential. So smooth muscle is also going to differ with skeletal muscle in a couple of ways. The first way is that the thick filaments are going to be fewer, and they have myosin heads all along the entire length. We went back and we looked at sarcomeres, and the sarcomeres have this Z disc with the thin filaments coming inward, and the other Z disc with the thin filaments moving inward. And then in between there you have your thick filaments. And the thick filaments are going to have these myosin heads only on the ends, and in the middle, where the age zone is, there's going to be no myosin heads here. But despite the fact that we don't even have a sarcomere arrangement, we do have thick filaments, and the thick filaments are going to have myosin heads all through the length. So the myosin heads are going to go all through the length of the thick filament, and then you have your thin filaments on either side. So we still have the thick filaments with thin filaments, but they're going to be fewer thick filaments. Okay. In skeletal muscle, the actual ratio of thick filaments to thin filaments is 1 to 2. So for every one thick filament, there is 2 thin filaments. Recall that in a myofibril that you do find in skeletal muscle, when you look at it in cross-section, you're going to have thick filament, and around each thick filament, you're going to have a total of 6 thin filaments, and they're arranged in a hexagonal pattern. Okay, so how does that translate to one of these for every 2? Well, there's another thick filament right here, and then it's going to have its 6 thin filaments. And so if you went through the entire myofibril and counted the number of thick filaments to thin filaments, it actually turns out to a ratio of 1 thick to 2 thin. But in smooth muscle, we're going to have fewer of those thick filaments and a lot more thin filaments. The ratio would be 1 to 13. And again, we don't have the arrangement of sarcomeres. There's no Z discs, but we'll take a look at how that actually works. Okay? So again, those thick filaments are going to have myosin heads the entire length, and that's not like what you see in skeletal muscle. So when we look over here, what exactly are the thin filaments attached to if they're not attached to Z discs? Well, if we went back to this picture from before and you looked at the smooth muscle in there, and we looked at some of the structural features in there, there's going to be these structures called dense bodies, which are proteins, and those proteins attach to this sort of lattice-like network of intermediate filaments. And so those intermediate filaments attach to those dense bodies. And it's those dense bodies that the thin filaments are going to be attached to. So in skeletal muscle, thin filaments are attached to the Z discs, and they end up pulling on the Z discs, shortening the sarcomere. But here, if we attach thin filaments to one disc and then to another, and then those thin filaments are pulled in, then the dense bodies get pulled towards each other. So if all these dense bodies get pulled towards each other, that overall shortens the smooth muscle cell. So we take a look at here, you can see that arrangement in this picture here, drawn more simply here. Here you have a dense body and then another dense body right here. And from the dense body, you have these thin filaments coming off away from there, and then you can see the red are those thick filaments with the myosin heads. So the myosin heads will pull on the thin filaments, and that would cause the dense bodies to be moving towards each other everywhere you have that arrangement of myofibers. So overall, you end up getting a contracted smooth muscle cell. So looking more closely, in relaxed up here, you have the relaxed cell, and then down here, when the myofilaments slide past each other and the distance between the dense bodies shortens, then you get the shortened muscle cell or the contracted muscle cell. Another way that smooth muscle differs from skeletal muscle is that we do not have the regulatory proteins that we see in skeletal muscle. Skeletal muscle has troponin complex, which includes the troponin and the tropomyosin. The tropomyosin is what gets in the way of cross-bridging, so the myosin heads cannot connect to and pull on the thin filaments. So we don't have that as a means of regulating contraction. Instead, smooth muscle has another regulatory protein called calmodulin that binds with the calcium. And we'll see how that works or looks here in a little bit. The thick and the thin filaments, these thick and thin filaments, the myofilaments, are going to be arranged diagonally. So when the thin filaments slide past the thick filaments during contraction, that's going to cause your smooth muscle cell to shorten and constrict or contract, but it's also going to pull the cell in such a way that it looks like it's been twisted. So that's why it says like in a corkscrew manner. And again, it's those dense bodies that are being pulled on by the thin filaments. So then we can say that these dense bodies that are right here are analogous to the Z discs in sarcomeres of skeletal muscle, since the thin filaments are pulling on them. And overall, when the cell shortens, the cell's going to actually bulge outward between those intermediate filaments, and that's going to make the cell look puffy overall when it's been contracted. So now the mechanism of contraction for smooth muscle. The mechanism that the smooth muscle goes through ends up showing that the muscle would be slow and synchronized in the way it contracts. And here, the cells are going to be electrically coupled with gap junctions. So over here I found this diagram here that shows two smooth muscle cells side by side, and the gap junctions are going to connect the cytoplasm from one cell to another. So when the signal and the action potential to get this smooth muscle cell on the left here to contract, it passes on that signal on to the next muscle cell. So as this one's contracting, it's getting the one right near it to contract right away. So your action potentials are transmitted from one fiber to another through those gap junctions. You can also see some structural connections that hold and bind the cells together. So as they pull on each other, shortening the entire sheet of smooth muscle. Some of the cells are also self-excitatory, and that means that they're going to depolarize without stimulation. So they have this regular intervals at which they automatically depolarize and repolarize for their action potentials. and in that sense that this muscle can act like pacemaker for those sheets of muscle. And cardiac muscles kind of behaves the same way. There's special structures within the heart that serve as pacemakers and are self-excitatory, depolarizing and repolarizing in a rhythm. But the pacemaking itself can be modified, okay, both in the rate and the intensity for their contractions. And this can be modified with the input from neural and chemical stimuli. So a nerve fiber can modify the rate at which these sheets of muscle contract, and in a sense is modifying the rate at which the pacemakers are depolarizing and repolarizing. So looking at the contraction of the smooth muscle, it can be similar, but not the same as skeletal muscle. And the following ways are what make smooth muscle similar. We're still going to see actin and myosin, and the actin will be sliding past the myosin as the myosin hits pull on the actin. So the same types of proteins are involved in contraction, so the same contractile proteins. We also see that the final trigger for causing the contraction is the increase in intracellular calcium levels. So we have to let calcium in to flood those the area where the myofilaments are. We also see that ATP is used to energize the sliding process, and the way we're going to stop contraction is by removing calcium. So we have to remove calcium from the cytoplasm to stop the contraction. So how is smooth muscle different than from skeletal muscle? Well, for skeletal muscle, all of the calcium comes from the sarcoplasmic reticulum, releasing it into the sarcoplasm. But for smooth muscle, because smooth muscle does not have well-developed sarcoplasmic reticulum, most of the calcium is going to come from extracellular space. That means from outside the cell. So if we look at the diagram over here of smooth muscle, this would be the membrane, the outer membrane of the smooth muscle cell, and this is your extracellular fluid. And in the calveoli I mentioned earlier, you have these calcium gates that are sensitive to voltage changes, and so when the action potential comes by, the gate opens and calcium comes in. So most calcium comes from there, but some calcium also comes from over here in the sarcoplasmic reticulum into the area. Now when the calcium comes in, it's not going to bind to troponin because we don't have troponin anymore. Instead we have calmodulin. So the calcium then binds to the calmodulin. So you can see when the calcium enters the cytoplasm or the sarcoplasm, you have your inactive calmodulin, but the calcium binds to the calmodulin, activating it. So now we have activated calmodulin. And then that activated calmodulin is going to go and activate myosin kinase. Now kinases are going to be enzymes that basically phosphorylate. So they transfer phosphates onto other molecules. Well, what is the specific name of this kinase? It's myosin light chain kinase. Recall that when we're talking about a myosin molecule, we're going to have the heavy chains that twist up, and the light chains make up the globular heads of the myosin. So here we see that when the calmodulin activates the kinase, the myosin light chain kinase, the activated kinase then goes and phosphorylates the head. Here we have an unphosphorylated myosin head, and once we activate this, the ATP now phosphorylates the head and activates the contraction now. So now the myosin heads can bind to the thin filaments and pull on the thin filaments to in that same kind of cycling that we see for the cross bridge formation in skeletal muscle. So here we see ATP is still needed to power this process. The big difference here is you don't have troponin and tropomyosin blocking the active sites for the cross bridging to occur. So we have that different regulatory system that involves the calmodulin and then another enzyme protein called the myosin light chain kinase that's doing the regulation. There is no tropomyosin blocking the way. So stopping the contraction is also a little bit different. It's going to require more steps, and so the process will be longer. So in that sense, the rate at which a muscle smooth muscle contracts and relaxes is going to be more slow. And the reason for that is more steps are required to remove calcium. We have to detach calcium from the calmodulin, and then we have to actively transport the calcium out of the cell because most of it came from out of the cell. And of course pump some of it back into the sarcoplasmic reticulum, but most of it we have to pump out of there. And then we also have to dephosphorylate the myosin itself. So here we have more steps required, and this takes it longer. So in that sense, because of that, the smooth muscle is longer to contract and longer to relax. The efficiency of smooth muscle contraction turns out that these slower contractions that the smooth muscle is reflected by the fact that smooth muscle is more efficient when it does contract. So this actually reduces the energy cost. So there's little energy cost to do this. And part of the reason is that the ATPase activity. So recall that on the myosin we have to split ATP. We have to take ATP and hydrolyze it to form ADP, adenosine diphosphate, and that phosphate has to go onto the myosin head. Well, the enzymes that are associated with that are called ATPases. So the enzymes that are doing this are actually slower. So you're going to get a slower cross-bridge cycle out of these heads. As these heads attach, pull, and then release, the whole process is going to be slower because the enzymes are slower. Another thing that smooth muscle can do is that the myofilaments, the attachment, the cross-bridge attachment, these filaments can latch together and hold on to each other for a period of time, and that can also save energy. And overall, most smooth muscle is going to maintain a moderate degree of contraction in there without fatiguing, and this is going to be referred to as smooth muscle tone. And so these the smooth muscle always has some level of slight contraction in there. And as far as ATP production within the cell, it's going to be aerobic respiration requiring both oxygen and mitochondria. And recall that from our earlier lessons that aerobic respiration is actually more efficient. It can produce a lot more ATP for the cell. The regulation of contraction can involve several aspects, including nerves, nerve fibers, messaging the smooth muscle. Hormones can also influence, as well as local chemical changes within the tissue fluid or interstitial fluid. So looking at neuroregulation, nerves are going to communicate with the muscle by neurotransmitters. And these transmitters are going to bind to the membrane of the smooth muscle, and they can cause either small local potentials or full action potentials, which are either small excitements of the membrane or great excitements. And if we get full action potential, all those things are going to do is increase calcium inside the cell. So the inside the cell, remember the cytoplasm specifically is called the sarcoplasm. We increase calcium in there, and that would get the concentration to go. Now, response depends on the neurotransmitter. So here, unlike skeletal muscle, you only have one neurotransmitter, acetylcholine, and the only response is excite the membrane and get a contraction. Okay. But here in smooth muscle, the response depends on which kind of neurotransmitter we're using, and as well as the type of receptor. So a same neurotransmitter can have a different effect on a smooth muscle in one place or one organ and different in another area or another organ in another spot. So both the neurotransmitter and the receptor can provide different responses. And what I mean by different responses, well, one is a response is to contract, and another one is not contract or relax. Okay. So that's what this next statement is talking about. One neurotransmitter can have a stimulatory effect causing contraction in one organ, and a neurotransmitter in another organ can have an inhibitory effect in a different organ. Again, and we're talking about the smooth muscles of those organs. So that's what's different. In smooth muscle, you only get, when the neurotransmitter is released, acetylcholine, you get contraction, but that's not the case with smooth muscle. It depends. You can get excitement and stimulation and contraction, or you can inhibit that process. Hormones and local chemicals also influence this here. So some smooth muscle cells are not going to have any nerve supply. So then how are they that they are going to contract? One is that pacemaking ability. Remember that some smooth muscle can depolarize spontaneously with a pacemaker action. Or they can depolarize, not spontaneously, but in response to some chemical stimulus. And the chemical stimulus, again, could be a hormone or other chemicals. And these chemicals basically are going to bind to receptors on the membrane, and the messages will be sent inward by way of a G-linked receptors. So that's the receptor is connected to a G protein on the inside. This sort of transduction pathway where we send the message into the cell was covered in previous chapters. Other chemical factors include, or the chemical factors that we're talking about can include hormones, carbon dioxide, changes in pH, and oxygen levels as well can stimulate a response here for the smooth muscle. Smooth muscle can respond to, some smooth muscle can also respond to neural as well as chemical stimuli. So in some cases, there is no nerve supply and it's depolarizing spontaneously or with chemical stimulus, as mentioned above. And in some cases, other smooth muscle in other areas of the body can respond both by neural supply or nerve supply and or neurostimulation and chemical stimuli. There are some special features for smooth muscle contraction, and one of them is the response to stretch, and the other special feature is length and tension relationships. So for the stretch relaxation response, the smooth muscle can respond to stretch briefly. In that case, sort of resist stretching, but then be allowed to stretch. And what this allows is the smooth muscle retains its ability to contract on demand, even though it may be stretching. And this is good for organs that do need to stretch, like the stomach. If you eat a big Whataburger or a big meal, right? Or your urinary bladder when it fills with urine, you need to be able to stretch outward. Yet the smooth muscle still needs to be able to contract to push the contents out of the organ. And then length and tension. There was this relationship for skeletal muscle where the skeletal muscle can provide a decent tension or pulling force if the skeletal muscle cells were not stretched beyond 120% or allowed to become shortened to more than or less than 80% of their resting length. So between 80 and 120% of resting length for skeletal muscle, you get optimum tension. For smooth muscle can change its length significantly more. Instead of 80 to 120, smooth muscle can become 50% of its normal length and can be stretched out to, it says twice here, well, that's equivalent to 200% of its resting length, and still provide good tension when it contracts. So what is this? What is the advantage here? This allows those organs like a stomach or a urinary bladder to be stretched significantly without becoming flabby, because if we stretch out to 200%, you can still get good contraction out of that muscle. So now we're going to look at the types of smooth muscle. And when we look at smooth muscle can vary in different organs by the following three items here. For the first one here, your fiber arrangement and organization can vary. So how the individual smooth muscle fibers are arranged and organized. They can vary by innervation, how much innervation or no innervation, and responses to various stimuli. So all of this depends on which organs you're at. That being the case, all smooth muscle can be categorized into two groups. It's either unitary functioning and as a unit, or multi-unit in which you have multiple types of motor units where the individual smooth muscle fibers need to be stimulated to contract rather than one contracts, they all contract. So looking at unitary smooth muscle, this one is commonly referred to as visceral muscle. Now, viscera is a general term to refer to internal organs. So here we're talking about all of your hollow organs except the heart. So remember, the heart has cardiac muscle. Organs that have unitary smooth muscle would have, the smooth muscle in there would have these characteristics. They're going to have opposing sheets of muscle, which is the longitudinal and the circular sheets. Remember, the longitudinal will shorten the organ, whereas the circular smooth muscle that goes around the organ would decrease the size of the hollow space or the lumen inside the organ. This type of smooth muscle is going to be innervated by varicosities, the unitary is. It's often going to exhibit spontaneous action potentials. In other words, has pacemaking ability. And individual smooth muscle cells are going to be coupled by gap junctions, which means if we get an action potential in one smooth muscle cell, then the ones next to it become excited to contract. And this type of muscle can respond to various chemical stimuli in there. So that's for unitary smooth muscle. Now looking at multi-unit smooth muscle, where can we find this? This can be found in the airways of the lungs, in large arteries, the erector pili muscles. Those are the ones that give you goosebumps. And in the iris of the eye, controlling the size of your pupils to either let more light into your eye. This type of muscle would have few gap junctions and little to no spontaneous depolarization here. So that would be rare. So no pacemaking, okay, within these sheets. So no spontaneous depolarizations. So how then, if we think about it, this type of multi-unit smooth muscle is similar to skeletal muscle because you're going to have independent insulated muscle fibers. So the independent fibers, each fiber must be stimulated individually to contract. This is not like the unitary. In the unitary, one contracts, the one next to it contracts, and the one next to that one contracts because of the gap junctions. This smooth muscle is also going to be innervated, but with the autonomic nervous system. And this innervation, these fibers are going to stimulate different smooth muscle cells within the smooth muscle, and basically these nerve fibers would form motor units like what we saw in skeletal muscle. Remember, it was in skeletal muscle one neuron for a certain number of muscle fibers. So if that one motor unit or nerve cell sent a stimulus, then only the muscle cells that were connected to that neuron or synapsed with it were going to contract. Well, the same thing would happen here in multi-unit. So you have basically a motor unit structure here. And you can get graded contractions, so you can grade the amount of tension in that muscle by recruiting more motor units, and that's similar to skeletal muscle. But how is this actually different? Well, it's out of our control. This is the autonomic nervous system. And another thing that's different is that the smooth muscle here can respond to hormones, and that's not true of skeletal muscle. Skeletal muscle only responds to a neurotransmitter from a motor neuron.